feelings and ability to read other people's integrity and future intentions would plausibly have been an advantage as well. Hypothesis 5 is that greater financial success led to greater reproductive success. Darwinian natural selection is fueled by some people with some organisms with some traits having more surviving offspring than organisms with other traits in the same population. This is, of course, not true today, where it's indeed in Europe and the United States, wealthier people have fewer children. But it was true in most of the world before what's called the demographic transition, beginning in the 18th century. And there are many studies that show that all over the world in pre-industrial societies, wealthy people did have more surviving children. And there's some evidence that this was true of Ashkenazim in medieval and early modern history. In his book, The Jews of Poland, B.D. Weinrib said that more children survived to adulthood in affluent families than in less affluent ones because of nutrition, less crowding, availability of wet nurses, ability to move quickly if there was a local plague, and other causes. And in general, this correlation until recently was certainly plausible. I call it, let's call it Tucker's Law after the famous quote of Sophie Tucker. I've been poor and I've been rich. Rich is better. Hypothesis 6. Many of the common Ashkenazi genetic diseases are a result of natural selection rather than genetic drift. Now, this is an important part of their argument and one of the more controversial ones. Biologists distinguish between genetic drift and natural selection as two possible causes of evolution, namely change in gene frequency over time. Natural selection, I've already explained. Genetic drift comes from the fact that in a finite population, there's going to be random sampling of which genes are represented in the next generation, having nothing to do with the advantage or disadvantage of that gene on the organism's phenotype. So let's say in a particular group of people, many of the redheads get struck by lightning, and so the next generation there's no one with red hair. It's not that red hair was disadvantageous. It wasn't that it attracted the lightning. It's just that stuff happens. Statistically, it may be organisms with a particular gene that don't do as well or do well, and gene frequencies are known to change as a result. This depends crucially on population size. As you can imagine, in a population of 200 million people, it's very unlikely that all the redheads will be struck by lightning. But in a population of, say, 5 or 10, that's not such an unlikely event. In particular, you see genetic drift in the form of founder effects. Imagine, let's say, Gilligan's Island, a very small number of people colonize an island. Let's say that they are never rescued and they are the founders of a new population. Well, whatever traits happen to be present in, say, the professor and Ginger would then characterize the population of that island several hundred years hence, having nothing whatsoever to do with the advantage of those traits, just the luck of the draw of who came first. This can also happen in population bottlenecks when an initially large population is decreased because of disease or warfare, and the particular traits of the people alive at a given time who then are progenitors of the ensuing populations, if there's rapid population growth, can also influence the genetic composition of the population. Kind of like if everyone is voted off the island, except a couple of people, then their traits will characterize the population thereafter. So CHH proposed 4 lines of evidence against what is probably the dominant hypothesis about the origin of common Ashkenazi genetic diseases, namely that they are consequences of genetic drift, perhaps in a founder population, perhaps as the result of a bottleneck. So the key assumption in testing these 2 hypotheses is that drift, by definition, affects all genes equally. What happens to the neutral genes, what happens to the advantageous genes, what happens to the disadvantageous genes is something that is not affected, by definition, by the process of drift. So their first argument is that Ashkenazim do not have less overall heterozygosity than much larger neighboring populations, such as Russians or mixed Europeans. So the idea is that if your population, your ancestors have gone through a population bottleneck, a lot of the genetic variation that existed beforehand will be lost. that therefore, as the population rebounds, the smaller number of genes that were around during the bottleneck will have many, many copies, but many copies of a smaller number of different variants. Therefore, it's going to be more likely that two identical variants will find each other from a father and a mother, and therefore, if a population has undergone a bottleneck, you'll have less heterozygosity. The claim is that Ashkenazi Jews do not have that reduced heterozygosity compared to much larger populations. Therefore, they have not undergone a population bottleneck, and the control would be small populations like the Samaritans, where they're so small that you really would expect genetic drift to decrease heterozygosity, and indeed in CHH's analysis they do have less heterozygosity, so it's not as if the method is insensitive to these effects were they to occur. The second argument is that Ashkenazi neutral genes are, as a statistical aggregate, close to those of mixed Europeans, and are as similar to mixed Europeans as the Russian sample is. And they claim, as the result of some fairly complex simulations, that this is not compatible with a bottleneck leading to a high frequency of deleterious mutations in Ashkenazi populations. That a gene that does nothing, that hurts you, as of course a gene like Tay-Sachs would, is unlikely to have reached a frequency of, say, 2% if there was, given what we know about how similar Jews are to neighboring populations. I should add, though, that tests for, this is my own editorial comment, the tests both for and against bottlenecks, from an outsider's perspective, do seem to be sensitive to initial assumptions and could go either way, and that this is going to be a matter of controversy between CHH and their critics for some years to come. The main argument, though, is that Ashkenazi genetic diseases cluster in a small number of metabolic pathways. That is, disease-causing genes scattered across the genome in different parts of the chromosome or in different chromosomes, by an amazing coincidence, cause diseases in the same chain of biochemical events, and hence lead to similar or disorders of the same metabolic pathway. Moreover, that for a number of these individual diseases, a number of different genes have arisen in Ashkenazi populations that all lead to that disease, and that this is unlikely to have occurred through chance. That if there's a common denominator of a single chain of biochemical events, and all the disease genes, or a lot of them, seem to affect that pathway, it argues that it wasn't just the luck of the draw of where you were on the chromosome that determines that caused a gene variant to arise, but because they all had a converging effect on a given metabolic process. So an analogy is that we know that in regions of the world that were host to malaria over the last few thousand years, a number of different genetic diseases arose, all of them involving hemoglobin, which is the molecule that the malaria parasite likes to latch onto, and so very different genes that all affected hemoglobin all led to variants that conferred malaria resistance. So the best examples in the case of the Ashkenazim were the sphingolipid storage disorders. Sphingolipid means sphinx-like fat. It's found in cell membranes and also found in the myelin sheath in neurons. You've all heard of the difference between gray matter and white matter in the brain. White matter is white because there is a fatty sheath that insulates the axons and allows neural impulses to travel more efficiently. Sphingolipids are one of the major components of that fatty sheath. It happens that in 4 of the diseases that disproportionately affect Ashkenazim, Tay-Sachs, Gaucher, Niemann-Pick, and mucolipidosis type 4, these are all diseases in which too much sphingolipid gets stored, leading to malfunction, even though genetically they're very heterogeneous. A second cluster are in diseases of DNA repair. When your DNA unzips and gets replicated, often there'll be copying errors. There are complicated mechanisms that repair those, basically repair the zipper when it breaks. They include BRCA1, BRCA2, Fanconi's anemia type C, and Bloom syndrome. All of them involve disruptions to this DNA repair mechanism. In the case of BRCA1, the acronym comes from breast cancer, and these are diseases that predispose people to women to breast cancer. But since all of them affect the same molecular pathway, they argue that it's unlikely that it was a coincidence that all four of these mutations, or set families of mutations, should have increased in frequency among the Ashkenazim. Now, how do you know that they're not just kind of cherry-picking diseases that happen to involve the same metabolic pathway after the fact? Well, in order to get a more quantitative estimate of this, CHH examined a functional genomic database, that is a database of all known human genes and, but more importantly, what they do, so that genes that are involved in the same metabolic processes are grouped together. They developed a computer simulation that randomly and repeatedly sampled 21 genes from this database, and they got a distribution of how often you'd expect randomly selected genes to be involved in the same metabolic process. They estimated then the likelihood, under an assumption of random sampling, that you would see the actual clustering observed in 21 Ashkenazi disease genes. That is, whether what the probabilities of obtaining that degree of clustering or a greater degree of clustering. And the claim is that the actual clustering is extremely improbable. Hypothesis 7 is probably the most important, which is that the recessive disease genes, Tay-Sachs for example, should boost intelligence in the heterozygotes, in the carriers. And for similar reasons, the dominant disease genes, such as torsion dystonia, should boost intelligence in the non-penetrated bearers. Well, there is an alternative hypothesis that even if you accept the hypothesis 6, that there was a history of selection rather than drift as an explanation for the prevalence of these genes, they may have been selected for something else, such as resistance to infectious disease. And indeed, CHH note that some diseases common to Jews, such as cystic fibrosis, almost certainly were defenses against infectious diseases, perhaps cholera, perhaps typhoid. They argue, however, that for most of the Ashkenazi mutations, there's no evidence that they have any conceivable purpose in resistance to infection. Moreover, that the European neighbors of the Ashkenazim suffered from the same infectious diseases, but don't have those mutations. They argue that the sphingolipid storage diseases might be a byproduct of the fact that a moderately increased availability of sphingolipids might increase, is known in vitro to increase the growth of axons, that is the output fibers of neurons or brain cells, and can also increase the growth of dendrites, that is the input branches in brain cells, and this is different sphingolipids have been shown to have this effect in studies of the brain development in mammals. That the BRCA1 gene, based on complicated statistical tests, can be shown to be a product of recent selection, and they speculate, based on the role of the normal version of the gene, that it might favor the proliferation of neurons early in brain development. And it must be noted that these are based in experiments on rodents. They're often in vitro, that is the growth of neurons in experimental preparations. The relevance of this to human IQ is, needless to say, highly speculative. What about IQ in humans? That would be the more direct test. Well, they cite 4 phenomena. One of them is that a number of studies of torsion dystonia show a 10-point IQ advantage over the controls. Torsion dystonia is a disease of uncontrollable muscle spasms that often leads its sufferers to spend their lives in a wheelchair. But people who have the gene but not the disease, according to a number of studies, have elevated IQ, although I must say the samples are rather small. That in non-classical congenital adrenal hyperplasia, where your adrenal gland produces too high a dose of hormones, 7 studies show higher IQ in the people who have the mild forms of the disease. And that in Gaucher patients in Israel, where because of the nationalized healthcare system there can be an exhaustive collection of all of the sufferers of the disease, They found 15% of the sufferers were either scientists or engineers, compared to no more than 2% in the population, and 32%